Okay, so this chapter is only 2 sections long. First part we're going to talk about meiosis, and the second part we're going to talk about sexual reproduction. So this should be loads of fun. Meiosis is very similar to mitosis, but also has some pretty distinct differences. So the goal would be for you to have to focus on the differences because you're all pros at mitosis. However, your tests say otherwise. So you might need to be pulling some big time double duty on the material over the next week. So, sexual reproduction involves both meiosis and fertilization. The good thing about sexual reproduction is that it introduces genetic variation into the offspring. That means you are not the same as your parentals. This accounts for the evolutionary success of sexual reproduction. Your gametes are haploid cells. Quick, remind me what haploid means. One N, but what does that mean? Half of what? Has half the number of chromosomes. Good. A zygote then is diploid, which means it has 2N, or twice the number of chromosomes. A reproductive cycle requires diploid cells to reduce its number of chromosomes by half in order to produce the haploid gametes. This process of reducing a diploid to a haploid is called meiosis. It's very similar in mitosis. It uses the same cellular mechanisms. The starting nucleus will always be diploid. However, all of your daughter nuclei will always be haploid. This is different than mitosis because mitosis starts with a diploid and produces a diploid. The chromosome replication is then followed by 2 rounds of nuclear divisions, where mitosis only had 1 round of nuclear divisions. So we call these 2 rounds either meiosis 1 or meiosis 2. So that's nice and simple. Before we can have meiosis 1, we do have to have interphase. Guess what? Lucky for us, it's the same interphase as mitosis has. G1, S, G2, and for a kind refresher for your little brains, G1 is where it focuses on growing the cell. S is where we have DNA replication. Each chromosome is replicated to produce 2 sister chromatids, which are held together with cohesion proteins at the centromere region. Then we have our G2, which is a final preparation for meiosis. Very similar. So, prophase 1 is by far the phase or section of meiosis that has the biggest and most differences than mitosis. So, in early prophase 1, the homologous chromosomes are going to be attached at their tips. And then this is done with nuclear envelope proteins. And then we eventually form this synaptomal complex, which is the lattice of proteins between homologous chromosomes. So this would be the little hair-like structures in here. Those are your complex that hold the 2 homologous chromosomes together. I kind of think of it as like the peanut butter of the peanut butter sandwich. Right, it's holding the 2 halves or 2 pieces of bread together. This forms at specific locations and then starts to spread outward to cover the entire length of the chromosome. So it doesn't, in the early stages, it's not the whole length of the chromosome that complex. Instead, it will start and then grow outwards. This creates a tight pairing between the homologous chromosome pairs. This is called the synapse. This is a formation of a close association between homologous chromosomes during prophase 1. This means that they're really held tightly together so they cannot be separated, which is super important because you don't want to lose a homologous chromosome in this process. And so when this peanut butter sandwich forms, the genes from the red chromosome will align precisely with the genes from the blue chromosome. So gene number 72 will line up with gene number 72, one on top of the other. Okay. This helps with crossing over. This is a phenomenon that only happens in prophase 1. and this is an exchange of genetic material between non-sister chromatids. So this would be like between the red and the blue chromosome in the picture. Not between a red and a red. It has to be between the two non-sister chromatids. This results in chromosomes that incorporate genes from both parents of the organism. It's how we get our genetic variation. Crossing over is one of two main ways to get genetic variation. Yes, wonderful. So recombination nodules, we're going to get a little bit more in depth here with this crossing over process than what we had in the past. And it starts with recombination nodules. And these are just protein bundles that form in that complex that are where points of crossover are going to occur. It's helped with multi-step processes of genetic recombination between the non-sister chromatids. So they're located at various intervals within that complex, and they have to be near a nodule. So what happens is, near a nodule, that double-stranded DNA of each chromatid will cleave. What does the word cleave mean? Yes, put that into a word. Chopped or cut. So the cut ends are then modified, and a new connection is made between the non-sister chromatids. So that would be between the red and the blue. There's a picture, I think, on the next slide of this. The complex then breaks down as prophase 1 continues to go on, and the chromosomes then begin to condense. Remember, at the beginning of prophase, you can't see them. They're not condensed enough. So they have to keep this condensing going on. Once the synaptin-mol complex is completely gone, the homologous chromosomes are still attached at the chiasmata. The chiasmata is, again, on the next slide, a picture of it, is where the crossing over events are going to happen. The number of chiasmata on a homologous pair can vary. There has to be at least one per chromosome in order for proper separation of the homologous chromosomes, but there can be as many as 25 in a single chromosome. So the next slide has some pictures. So here you have your homologous pair. And then this little circle right here shows you a chiasmata. So that's just referring to the point of contact between the homologous chromosomes. So another chiasmata would be up here. So that's where the DNA cleavage or cutting will happen. And what ends up happening is they really just switch pieces. Okay, and they are called recombinants. Oop, did you have a question?
No, I'm just...
So once, this is another diagram of crossing over if the whole color thing, Brett, isn't your jam. I have letters associated with it. So this blue chromosome gets the capital letters. The red chromosome has the lowercase letters. Here is your crossing over event. What is that location called? Chiasmata. Okay.
That was such a struggle.
Yes, it was. And so now you can see that the blue chromosome, which had all capital letters, now gets lowercase c, and the red chromosome, which had the all lowercase letters, now has a capital C. So this could be how the alleles separate. This is how you might end up with a dominant version, but somebody ends up with, or your sibling can end up with a recessive version. So prophase is still going on, but at the end of it, after crossover, that complex breaks down, the cohesion proteins between the homologous pairs are removed, so they can now condense, and the pairs are now only held together at the chiasmata. So it could be held at 25 different places, 3 different places, 1 different place, but the chiasmata is the only thing holding the homologous pairs together. And it is important for you to understand that they are connected. They are physically in contact. Okay? Yes. So the pairs are held together only at the chiasmata, and they are now called tetrads. Tetris, you know the game of Tetris? All those blocks are made up of 4 different They're rearrangements of 4 different blocks because tetra means 4, okay? Tetrads are 2 duplicated homologous chromosomes which contain 4 chromatids. You see all 4 chromatids in there? 1, 2, 3, 4.
Where'd you get 5?
All right. Crossing over. The last piece about crossing over. This is the first source of genetic variation. A single crossover event leads to reciprocal change of equivalent DNA. So what that means is if you look at this, the red and the blue that switched are the same gene and the same size. They're not 2 different sizes that got flip-flopped. It is 1/2, 2/1. It's that reciprocal idea there. Clayton would be proud. When recombinant sister chromatids are moved into a gamete cell, it will carry some DNA from one parent and some DNA from the other parent. Just recombinant, that's a word you're going to see a lot. These are the recombined versions of the gene. Anytime you see recombinant, it's the after crossover result, I guess. The recombinant chromatid has a combination of maternal and paternal genes that did not exist before crossover. It is important for you to understand this does not happen at fertilization. This has happened to your eggs and your sperm already. Okay? Actually, females with your eggs ripening before your cycle, it has to finish this meiosis process. Okay? Some meiosis has happened while you were still in the womb. The rest of it has to happen every cycle. Okay? So by maternal and paternal genes, it's referring to your parents, not you and your baby daddy. Okay? That happens with fertilization.
That's really gross.
Last thing about crossing over is that it can occur almost anywhere wherever those chromosomes are synapsed. There's no rules as to where crossing over can occur. Okay, there's no, this is part A. Crossing over doesn't happen at part A. That's not true. Crossing over happens anywhere where there's a synapse, or can happen anywhere there's a synapse. Finally, prometaphase 1, which is for some reason your guys' least phase of mitosis, is prometaphase.
We never learned about it when we were
I know, but it's super simple. The only thing that happens here is those spindle fibers hit the kinetochores. That's this in-between phase here. They're not quite at the metaphase plate. They're just connected to. And as a kind refresher, kinetochore proteins are a multiple protein complex that bind to the centromeres, to the microtubules of the mitotic spindle. The microtubules will then grow from this microtubule organizing center, which we call centrosomes. At each pole, they grow towards the middle of the cell. They will then attach to one of the kinetochores of the 2 fused homologous chromosomes. Each homologous chromosome will take a microtubule from the opposite pole, so it gets pulled to each opposite pole. Pull, whatevs. Metaphase 1, the homologous chromosomes arrange at the metaphase plate with kinetochores facing the opposite poles. This is a new piece, but I think it's super interesting. The homologous pairs will orient themselves randomly at the equator. So that means if we have our homologous pair, it can either line up green-blue, or it could line up blue-green. Okay, it doesn't have to be one way or the other. So each pair has 2 possible alignments. A, B, B, A. Green, blue, blue, green. Does that make sense? And so this randomness of the alignment at the metaphase plate is responsible for more genetic variation. So this is genetic variation thing 2. The first is crossing over, which gives genetic variation. And this randomness of alignment is the second thing that gives genetic variation to these cells. The let's see, each orientation of each tetrad is independent of the orientation of the other 23. Well, humans would have 23, but the other tetrads. So just because the first tetrad goes green-blue doesn't mean the second tetrad has to go blue-green. It could go green-blue, 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 green, blue, blue-green. They don't affect one another. So, like I said, they each have a possible arrangement, 2 possible arrangements at the metaphase plate. This gives the possible number of possible alignments a value of 2n, meaning 2 to the number of chromosomes. So n is the number of chromosomes per haploid set. In humans, this would be 2 to the 23rd, which if you throw that in your calculator will be over 8 million possibilities of genetic recombination simply based on how the chromosomes line up at the metaphase plate. This does not include the possibilities when you factor in crossing over as well. Okay, I think once we add that in, it's over 16 trillion possibilities. Yes, it is insane. So this kind of shows it to you in picture form, what I just talked about. They're using blue and red. So option 1 would be blue, red, the lineup at the metaphase plate. Option 2 would be the 2 blues are not on the same side anymore. So then it shows you what the possibility of the gametes would be as you go through. So 4 different possibilities of the gametes then. Anaphase 1, the microtubules are going to pull the linked chromosomes apart. The sister chromatids still remain tightly bound at the centromere, and the chiasmatas are finally broken in this phase. All right then, Faith. I feel like these two are like high-fiving and kicking at each other. Yeah. Or isn't it like one of those inflatables where you put like the bungee on and you run and you get like pulled back and it pulls back at where the strap is and your legs and arms kind of still are flailing forward? That's what I think of when I see that too. Telophase 1 in cytokinesis, the separated chromosomes will arrive at opposite poles. The remainder of typical telophase events may or may not occur depending on the species. So some organisms, the chromosomes will decondense and nuclear envelopes will form. Other species, cytokinesis will happen without the formation of new nuclei. In animals and most fungus species, you'll get a cleavage furrow, cells. Plants will have a cell plate show up. This always will result in 2 haploid daughter cells. However, this is, when you think about this, your chromosomes are still going to be sister chromatids. Okay, so in mitosis, we called this still a diploid cell. However, since this is meiosis, these are considered haploid because only 1 full set of chromosomes is present. Okay, because half they split. Okay, so even though these look diploid, they're really only considered haploid. Then we get meiosis 2. Some species will have an interkinesis. This is the brief period of rest between meiosis 1 and meiosis 2. This is sometimes for regrouping. It does not have an S phase. We cannot duplicate our DNA anymore at this point. The 2 cells that were produced in meiosis 1 will go through meiosis 2 in synchrony. What happens to one happens at the same time for the other. Same, synchronized swimming, everything happens at the same time. The results of meiosis 2 will be 4 daughter cells with half the number of sister chromatids. So what does it look like? Very much so like mitosis. Prophase 2, if the chromosomes decondensed, they'll recondense. If there was a nuclear envelope that formed, those will break down. The centrosomes move towards the poles, and the new spindle fibers will form. Prometaphase, same song, different dance. Nuclear envelopes are completely broken down, the spindles are fully formed, and the microtubules attached to the kinetochores. Metaphase 2, your sister chromatids are maximally condensed, and they are lined at the equator, not equation, sorry, or the metaphase plate. And this shows you, the diagram shows you what anaphase looks like here in meiosis 2. The sister chromatids are pulled apart and moved to opposite poles. The non-kinetochore microtubules will then elongate the cells. Telophase 2 and cytokinesis, your chromosomes are at the opposite poles. They begin to decondense, and now we get nuclear envelopes forming, and this will result in 4 unique haploid cells. There's big diagrams. I tried to put them into this presentation, but they did not copy over well enough they needed to be like zoomed in so you could actually read them. So you should take a look at them in your book, and this shows you the connection between meiosis 1 and meiosis 2. I do like this chart because it's a good comparison between meiosis and mitosis. So that first row tells you the process, and then the bottom 2 tells you when and if it happens in either meiosis or mitosis. So DNA synthesis for both meiosis and mitosis occur in the S phase of interphase. Synapse of homologous chromosomes, that happens during prophase 1 of meiosis, and it doesn't happen at all in mitosis. Mitosis doesn't do homologous chromosomes at all. Crossing over happens during prophase 1, does not happen during mitosis. Homologous chromosomes line up at the metaphase plate. This happens during metaphase 1, but not mitosis, because mitosis doesn't do homologous chromosomes. It does sister chromatids. So the devil is in the details this chapter. Sister chromatids line up at the metaphase plate. This happens during metaphase 2 during meiosis and during regular metaphase of mitosis. The outcome, we get 4 haploid cells at the end of meiosis 2. And mitosis, we get 2 diploid cells at the end of mitosis. I think this chart is a fair detail, and it's very simple, and it's very doable. And I'm expecting you to know this information from this table.